So hi, hello everyone again. Microbehunter here. Welcome to another live stream, to another Saturday live stream. Today I'll be doing a little bit of microbiology, of course, also some microscopy. This camera on my desk is set up a little bit different today because I need a little bit more space. Because I'll be showing you how to make Petri dishes. With bacteria on it, and of course we'll be putting some of them also under the microscope. I hope that all of you are able to hear me. Usually at the beginning I do a little sound check, so maybe you can quickly type into the comments section, into the chat section, if you're able to properly hear me or not. Usually there is also a little bit of a couple of seconds of time delay. Yeah, sound is good. I can already hear. Back again. I'm here again back after 1 week of not doing a live stream. But today I would like to show you a little bit some introduction into some basic microbiology. There have been some requests for me to do this. I had some mixed feelings about this initially, also because of safety concerns. But today I think I can show you a fairly safe method because I found a safe source of bacteria. One thing that I would like to do also today is the following. As you're typing comments, please continue to do so. You can ask questions as well, of course. But what I would like to do is I would like to maybe not respond to the questions right at the beginning. I would like to keep the first part relatively compact. So I'm going to maybe ignore the questions a little bit at the beginning, and then later on in the second half of this live stream, I'll be going through the chat to respond to any questions or comments that you might have. Now, if you are new to this live stream, then the rules are as follows. Rules under quotation marks is of course you can also ask off-topic questions in the chat. I will try to answer them as well, but they do have to be a little bit topic related, in the sense it should be about microscopy and or microbiology. Okay. Another announcement that I would like to make is the following. I actually posted this also on my webpage. I would suggest the following. I don't know what you think about this. This guy here is Anthony van Leeuwenhoek. He is the first microscopist around, and I realized that he died. He passed away exactly 300 years ago. So I think this is a reason enough to celebrate 300 years of hobby microscopy, 300 years of microscopy, and therefore I suggest the following. It's as a community project, so to say. I would like to encourage everyone who is interested in microscopy and nature observation to make videos and or maybe write articles, or it doesn't matter, and then add the following hashtag to the content, hashtag microscopy 300 years. By typing this into the search bar, then we're able to find all of the videos and contents that have been made with this hashtag. So this means that even people who do not have a large or well-known YouTube channel, by making videos about microscopy and by adding this hashtag in the title or in the description, then it's easy to find all of them. And I would say that this could be a way to celebrate 300 years of microscopy. Just to make this clear, the videos will not be sent to me. You just publish them on your own YouTube channel. And if you have a Gmail account or a Google account, then you already have a YouTube channel and you can start uploading. Then we're able to find the videos. And I think this would be a very nice possibility to share a little bit knowledge about microscopy and to create a community. Yeah. So what I would like if you want to know a little bit more about this, I will make a separate video about this. But if you visit the microbehunter.com webpage, I actually put a link as well with some suggestions for videos. So this is going to be a completely decentralized project for anyone who's interested in participating. So what I will be what I would say is the following. I'm just going to get started now, and I'm going to also not only show you what I'm going to do, but I also would like to explain a little bit the background of why I'm doing the things that we're doing. And I'm not saying that if you're interested in hobby microscopy, that this is what I'm going to be showing you today is absolutely necessary to know, but rather I think it's simply an additional piece of information that you might find useful or not. And maybe some of you maybe at university, college level, might also find this useful. Okay, So first of all, what I will be doing today is the following. So a short overview. I'm going to show you how to grow bacteria on a petri dish. Number one. Number two, I will be putting them under the microscope. I'm also going to show you how to heat fix bacterial slides. And I will be also staining the bacteria with a methylene blue solution. Okay. And I think that's going to keep us quite busy. And yeah, I also do have a new stereo microscope standing next to me, so where I'll be showing you also some of the bacterial colonies here. So first of all, I'm going to a very brief introduction. Bacteria these days are grown in petri dishes. And those petri dishes, they have a lid, obviously that's very important. And in here, this is this colored material, that is the so-called the growth medium, which contains nutrients for the bacteria to grow. And this growth medium, there are hundreds, if not thousands, of different recipes for different purposes and for different bacteria. And this is a very gel-like substance. It's gelatin, but it's not gelatin, but it is yeah, like gel, a very solid gel. And the substance that is used to make this is known as agar. And it's also used for cooking. So some people, so I actually found this one over here. This is not laboratory grade agar, but it's used actually for making cakes instead of gelatin, because gelatin is a protein. It's from animals. And people who don't like that, they sometimes use agar, which is a plant-based polysaccharide, which has been used since the 19th century for making those agar plates. And those agar plates here that I've got here, I've got a few of them here that I'll be using. They are commercially made. So I actually bought them over Amazon, ready-made, you know, with the medium already in it. But normally what you would like to do is to actually make them yourself. And I've done so a few weeks ago, and I would like to show you now a video of how I've been preparing and making those agar plates myself. The agar plates that I use here, these are disposable plastic petri dishes, okay? And of course, there are also glass petri dishes available, okay? But we nowadays in microbiology, you kind of rarely use those because you want to use the disposable ones, okay? So what I would like to therefore do now is I'm going to start off with a video. And yeah, here you see myself making, turning on the Bunsen burner. And what I will do is I will occasionally pause it and I will show you what I'm doing. Oh, and I just see that in the background, you see that there is still my laundry over there. And what I will be doing just a second. So I think this looks much nicer. Okay. Yeah. So basically, what I would be I'm doing here right now is I'm making those petri dishes, and those petri dishes are packaged sterile. So this basically means that they're free of bacteria. And you have to open them, and the gas flame next to it is really important for making sure that no bacteria are able to fall into those petri dishes. Now you might wonder a little bit is how do the companies make it sterile and yeah, free of bacteria? And that is, they simply manufacture them, and then the whole pack, the box, is sent through a reactor, and it's being with radiation. And this radiation will actually kill the bacteria. So it is sterilized after it's been packaged, and because it's sealed off, therefore bacteria are not able to enter it. Okay, so let's move on here. So I'm carefully removing the petri dishes now from its sterile packaging, and next to it you can almost not see the blue flame of the Bunsen burner. And this is really important because the hot air of the Bunsen burner will create an airflow upwards. Okay, so there is this hot air rises, and it kind of pulls air along from the bottom. And this airflow, which goes upwards, prevents dust and bacteria, which can be found in the air, from settling down into the petri dishes. Okay, so that's actually a very important way of preventing bacteria from falling down, is by creating this airflow. Okay, so yeah. Next to it in the flask, you can see the culture medium that I prepared, which contains the agar and also yeast extract. And it's covered with aluminum foil, and the whole thing has been autoclaved. This means it has, I put it into a pressure cooker. and this pressure cooker kills off all bacteria. So it's heated up to over 120 degrees centigrade under pressure. And what I'm doing right now is I'm holding the top part of the flask over the gas flame to heat it up. And this will also create a hot air cushion. And when I remove the cover, the aluminum foil, okay, then because the top part of the flask is very hot, it creates a hot air cushion which prevents bacteria from falling into the flask, contaminating bacteria from the air. Yeah. So this is, and now I'm pouring it out. The agar nutrient medium is still warm. Yeah. And I am basically pouring it out. And then I have to wait for some time for the agar medium to turn solid at room temperature. Yeah. So this does require a pretty steady hand and patience. And every time after I've done this, I've reheated the top part of the flask to prevent bacteria from falling in because of this hot air cushion, so to say, that basically forms around the opening. So and that's basically finished. And then you have to wait for a couple of hours for it to cool down, and then you can store it in the refrigerator, and that's pretty much it. Okay, so now the video starts again, and here I'm back again. Okay, do you want to know the agar and the yeast extract and how does it look like? Well, the agar is just a powder. I'm just going to open this. Where is my here? Look, this is actually a slide box. This is a slide box that I'm now using also as a toolbox. Okay, I want to show you how the agar looks like. It's just a regular powder. Yep. It's really nothing spectacular. Yeah, it's powder. I mean, you're able to see it here, right? Just some white powder. Looks a little bit like flour, and you have to mix it with water, and then you heat it up, and then when it cools down, it starts to turn solid. Yeah, and another thing that I used is yeast extract. Yeast extract is not yeast. It's but it's the extract of yeast, and this is also dissolved in water. And then this serves as a food source for the bacteria. So what I've done is I used 15 grams of agar on 1 liter of water and 3 grams of this yeast extract. Yeah? This is not. I did not adjust the pH. Normally, you're supposed to do other adjustments as well, but I didn't do that. And but these plates that I'm using here right now, are commercial plates that I bought over Amazon. They're actually designed to grow fungi, but I was also able to grow bacteria. So this plate I already made. And now let's talk about bacteria. On YouTube, you're able to find lots of videos where people will actually grow their own bacteria from their fingers. So what they do is they make those agar plates, and then they basically, I'm not doing this right now because the lid is on, but then they're pressing their finger on the agar plate. And then you wait a few days and you're going to see the bacterial colonies growing on here. And this kind of shows how dirty your finger is. And it's a very impressive little experiment, but I highly disadvise you to do that. You do not want to grow bacteria at home that are able to grow on your skin. It's not safe. And then you're already in a higher safety biohazard category. If you want to work with, like in this demonstration here, I want to work with bacteria that are extremely safe. Okay, so you need a safe source of bacteria, and I'm going to show you now one, and you'll be surprised where I bought them. We got them from the drugstore. Yeah, they're capsules, medicine. These are actually E. coli bacteria. I don't know if you're able to read this here. It says here E. coli. Yeah, here, bacteria, freeze-dried. So people who have a disturbed digestion because maybe they have been on antibiotic treatment, which kills off bacteria, also those in the intestine, or people have constipation and so on, you can swallow E. coli bacteria, and they are can be found in capsules like this. Yeah, here. I've already used some of those, right? And in here, I'm going to cut them open. In here, there are freeze-dried E. coli bacteria. And what I'm going to do is I would like to simply grow those freeze-dried bacteria on a Petri dish. And then you have to wait 2 days. And because we do not want to wait 2 days, I've got a ready-made one over here as well. Okay, So this is basically the thing here. I'm just looking over here into the comments. Some people say, "Wow, E. coli in the drugstore." Well, I'm going to show you two other sources of bacteria. I bought this one here as well. You know what this is? Yeah, these are bacteria, freeze-dried bacteria that you can add to milk to make yogurt, homemade yogurt. Where do you buy this one? I also got it over Amazon. Yeah, so you can buy freeze-dried bacteria, safe ones, like the one over here, which you're able to eat, the E. coli that you're able to eat. These are actually, I think, 2 or 3 different types of strains, bacterial species mixed together to make yogurt. Yeah, so this is also readily available, and something I didn't even know. This is actually a co-worker who's got aquariums gave it to me. These are bacteria that you can add to an aquarium to call it probiotic bacteria for fish, because aquarium also need to have a certain bacteria in there, and you can add those as well. And you can, if you look into it, it looks like powder. Yeah. So, and it smells pretty bad. Okay. So I just wanted to show you that bacteria can be bought. Normally, if you are doing research, what you do is you write to an organization or company that sells cell cultures, and you can order bacteria, whichever ones you want, almost. Of course, but they're of course expensive, and they might not send it to private people, right? So, and therefore we got those from the drugstore. The reason why, that's a little bit the background info, is because I'm a teacher and we're doing some science experiments in schools, okay? Yeah. So this is kind of the idea, and I'm going to now show you a method of actually plating them. And in order to do so, or before I do that, I actually have the picture of 2 people here. It is this lady over here, yeah? Fanny Hesse. Yeah, she is actually the woman. She was the wife of a microbiologist, and she actually suggested to use agar as a medium for petri dishes back in the day, 19th century. Yeah, because at that time, the scientists, the microbiologists, they used gelatin. And gelatin is a protein and is not, you cannot heat it up very much. Okay, so that's a problem. So you needed something that is very heat stable. And then she suggested to use agar because she herself has been using it for making food, for jam and things like this. Yeah, and the agar was obtained from Indonesia from an algae, and ever since then, agar has been used in microbiology. And this guy over here, his name was Julius Petri, and guess what he invented? Yeah, he invented the petri dish. Okay, because at that time they had real problems when they were growing the bacteria open, then it got contaminated. So he basically said is we gotta have a dish with a lid on top, so to prevent of course also from the medium from drying out, and also from preventing contaminants from falling in. Okay, so this is basically 2 important people in the development of those agar plates. Yeah, that So she is often forgotten a little bit, but actually she's got in Wikipedia, she's got a pretty decent sized article, because this is was a very important contribution. As a matter of fact, Robert Koch, the important microbiologist, he actually then discovered the bacterium for tuberculosis using agar, by growing them on agar plates, okay? So what I'm going to do now is I'm going to use a pen, and I'm going to explain to you in theory right now what I'm going to be doing so that you understand better. I'm going to show you how to make a so-called a dilution streak. Okay, so I'm just going to use this here as a little so that's going to be my agar plate. And what I will be doing is the following. I'll be using this tool. It's called an inoculation loop, to pick up the bacteria and to transfer them onto the agar. And I'm going to show you now using a pen what I'm doing. I'm going to be dipping the inoculation loop into the bacteria, and I'm going to take a small sample, and I'm going to spread the bacteria on the agar like this. Okay, and then I'm going to hold the loop into the flame to kill off all of the bacteria. So there, what I'm going to have here is I'm going to have a very, very dense, we call this a bacterial lawn, very dense spread bacteria, and too many of them together. But what I want is I want to see individual colonies. So I hold the inoculation loop into the flame, and then I do the following. I go into the bacteria here, and I streak across, maybe twice, and then I spread again. So what I'm doing is I'm taking a few bacteria from over here, and I'm going to spread them over a larger area again. Okay, then I hold it again into the fire. Okay, and then I have to cool it down. So I'll be cooling it by putting the loop into the cold agar, and then I'm do it a third time. I streak across, and I will go like this. Okay, and so I'm diluting the bacteria. So what I will have at the end is I will have individual bacteria over here, but I will not be able to see them because, well, they are microscopic. But then I have to wait for 2 days, and this bacterium over here will start to divide exponentially from 1 to 2. It doubles 4, 8, 16, it doubles 32. You get the idea. It grows exponentially, and after about 24 hours or 48 hours, what I'm able to see is I'm able to see with the plain eyes, I'm able to see a colony over here, yeah, and over here, and over here, and over here, and over here. Okay, so this is kind of the thing that I'm going to be able, that I would like to see then at the end. And as a matter of fact, with this plate over here, if you're able, maybe I'm going to use a dark background here. Yeah, you're able to see how I have done that. Yeah, and you're able to see over here those individual dots that you see here. These are the individual colonies. So one of those dots here comes from one bacterium. Okay. And this is a very important standard procedure. The reason is because by looking at it now, I'm able to see that those colonies here, they all more or less look the same. They have the same color, for example, and therefore I can assume most likely I have a pure culture, which means that if only one got one type of bacterium. One thing that you are going to notice, however, that the size is not always the same. So why is it that, for example, the colony over here, they are so much larger than the ones over here? Yeah, I mean, if this is the same bacterium, shouldn't there be the same size colony? And the reason why they are so smaller, much smaller here, is because they're competing for the nutrients. They're running out of food, and they're building up wastes, and therefore they cannot grow as quickly as the one over here. Yeah. Because this is pretty much it has much more food available because it's further away from the others, yeah, and then the food is able to diffuse to it, and it's able to use it, and then it starts to produce wastes, and those wastes basically are also able to diffuse away more easily, yeah. So this is a little bit the idea, okay. And what I would like to do now is I would like to make such a dilution streak now, and because we cannot wait for 2 days, and we're going to then continue with this one here, and then I'm going to show you with this one over here. We're going to put some bacteria under the microscope, okay. I also see over here in the comments already that some people have petri dishes that are made of glass. Sure, that's indeed possible, but the ones that we now routinely use in a microbiology lab are disposable ones that are made of plastic. Okay, so what I'll have to do now is, and this is a little bit where something that I gotta be careful with, is I have to use a Bunsen burner. I have to move it away so that I don't burn myself. Okay, put the mouse away, I put all of the things away, and I will just also explain to you how you use a Bunsen burner. You've got to be careful here. This here is for the gas. Yeah. And over here, this one over here, if you're able to see this is the air supply. Yeah. And you're able to turn this, and then you're able to, the hole is closed. And now there is a lot of air. Okay. Is able to enter, and you see that the hole is open, and now it's closed again, right? This is pretty important for adjusting the right amount of air. And for lighting it, I always usually cut the air supply off a little bit. And when I light I have to turn it on. I'm getting a very yellow flame, okay? And then I have to turn the air supply on, and it will switch over to a blue flame, and that's actually what I want. I have to be careful because I've got a camera over here. I don't want to burn the camera. Yeah. So this is basically the first thing. Over here, I've wrapped it. I've closed them off using parafilm. I'll show you a little bit later. So I'm going to unpack the thing here. Something I did not show you yet is I have disinfected everything with alcohol. Got to be careful with alcohol and fire, okay? But everything's been disinfected with alcohol, so to make sure that there are no contaminants going in here. And what I'll be doing next is I will be opening next to the flame. I'll be opening one of those capsules, and I'm going to put out the freeze-dried bacteria here so that I'm able to use those. Okay, so I'm just going to take it out. I've also disinfected my hands using the alcohol, but you need a little bit of time here for it to also properly evaporate. Yeah? So, and I'm just going to cut it open. Yeah, and I'm going to put the freeze-dried bacteria, E. coli bacteria in here. Okay. Hopefully without making a mess. Okay. Yep. Okay. So that's basically, these are freeze-dried E. coli bacteria. I'm going to throw the capsule away. And I would now like to grow them. Okay. So the first thing that I have to do now is I have to disinfect the loop. And I do that by holding it into the flame and by glowing it. And I think those inoculation loops these days are also made of platinum and covered with platinum to kind of minimize corrosion a little bit. And now I have a problem. If I basically take this extremely hot loop and go into the bacteria, I'm going to kill those bacteria. Or I might set it on fire, right? So what I'm going to do therefore is I'm going to cool it off by putting the loop into the agar medium, okay? So I'm going to carefully open the plate, only, excuse me, only enough, I'm just going to barely open it, and I'm going to now put it here on the side, and it makes, okay? It's now cooled off, but free of bacteria. I'm now going to take some of the bacteria here, and I'm going to start streaking. And you know what I'm going to do now is I'm going to probably put the camera down a little bit more so that you're able to see it better. Okay. There's a little bit some condensation water here on the lid. Okay, so what I'm doing right now is I'm going to start streaking. You don't want to talk during the while you do that, because otherwise you're going to blow bacteria and air on it. And now I'm going to glow it again to kind of kill off all of the bacteria. You cannot see it now because the camera is pointing away. Okay. Yeah. So yeah, but you know what I'm doing. And I'm going to now cool it off again. Now I'm going to go into the streak, I'm going to pull over, and I'm going to spread it again. You always have to be careful that you don't scratch the surface. Yeah, I'm glowing it out again. Okay. Okay. Get the idea? Yeah. And then I put it again into the cold agar medium. And this also kind of cleans off a little bit the soot and the black, burned material. I go again into the previous one, a streak out, and I spread it again. And then, I do this again, and I'm finished. I turn off the gas. Okay, I put the inoculation loop away. And now, what you do is you turn the plate around and you label it on the bottom side. You never ever label it on the lid, because then you might exchange the lid. And as a matter of fact, this has happened. When I was at university, in a different microbiology department in a different country sent us some samples, and they were labeled on the lid. And then you already know that the person who did that was not properly trained. So normally, I don't do, oh gosh, here it is. So normally what I do is you put on the date. Today is what, the 17th of July, okay? 17th of July, 2023, and these are E. coli. E. coli bacteria. And I always label the plate on the side, because if I write across the center here, then I'm not able to see the colonies properly. So that's basically almost finished, because I'm going to show you now the coolest invention ever, and that's this one here. It's called parafilm. I love this. This is actually a fun part. I'm going to show you what this does. I think this is the coolest invention in the whole lab area. I'm going to cut off a little strip here of parafilm. And this is the protective paper covering that I'm going to remove now. And I'm going to go on the corner here. And oh, I forgot something, to do something. I have to label this here. I'm just going to make a blue streak so that I know which type of medium it is. Up here, now you see it, okay? So in the lab, we always used to color code the plates, so that because we had so many different agar plates of with different recipes, we color coded them, right? And so that's also something I'm gonna do here, simply to keep up the tradition a little bit. And now I'm going to do the following. Look, I'm going to hold this here, and when you stretch it, it pulls out, and it starts to become sticky. It starts to stick. Okay, and this is important to prevent the Petri dish from drying out. And and that's it. And now I'm going to wait for, oh, the thing went off, unfortunately. So the 17th, okay. And I'm going to wait now for 2 days, and then I'm able to see what am I able to see after 2 days? Well, this one over here. Okay. The plate that I showed you, and you can see that I made this on the 13th. So let's go again here a little bit, and you're able to see that the individual colonies are visible. 17th of June, not July. That's correct. Somebody reminded me. 17th of June. We're still. I'm 1 month off, of course. So what's the point of properly labeling a plate if you put on the wrong date? 17th of June. Yes. So that's basically how it will look like in 2 days. 2 days later, and now we can do with this here whatever we want to do. Okay. And I'm going to now show you how to put it under the microscope. Okay. And I'll be using this plate over here. So normally you also, again, of course you have to again turn off on the flame. And what I will be doing now is I'll be using a slide. Okay, so here a slide, and I'm going to now put the slide here, and I need a little bit of water. So look, I've got this pipette here, but I forgot where I put my tips. Where are the tips? The tips are in here. Okay, so in case you have not seen this either, the microliter pipette. You go in here, and here we go. And now I'm able to take up some water with this. I've prepared some water over here. Okay, and here is a drop of water. I'm going to, again, you just have to trust me on this, that there's a fire burning here. Okay, and go turn it down a little bit. And I'm going to now do the following. I'm going to take this plate over here, and I'm going to now pick off one colony, and I'm going to put the colony into the water. So it doesn't even have to be a colony. I can take some off from the side here as well. So I'm going to I'm holding it again into the flame. So let's cool it off again. Let's pick some material off here. Okay, I see that there is something on here. Okay, And I'm just going to suspend it. Yeah, on here. I'm going to glowing it again. Okay. Turn off the fire. And then what do I do now? Then I'm going to wait until the water has dry. And you actually see that there is something on here, right? Now, I've already got some dried slides. I prepared everything, of course, yeah. But because I would like to show you how to heat fix it. What I'm going to do now is I'm just going to put a cover glass on top here so that we're able to see it directly, just for the fun of it. But actually, I wanted to show you something different later as well. Yeah, you can, of course, keep posting comments and questions. If you are new or if you just joined, after I've done the theory part and the demonstration part, I will basically go through the chat, and I'm going to then answer some of the questions that you might have. So let's go up, let's put this in here, okay? And let's switch over to the microscope view. Okay, here we go. And I put it here under the microscope. Okay, I need more light, and the magnification is way too low, obviously. So 20 times. Yeah. Okay. 40 times. Yeah, the bacterial density is quite high. And you might be a little bit disappointed. At least I hope that you're disappointed, because the purpose is to actually show you how much better it will look like after you've dried it and heat fixed it and stained it. That's actually the whole purpose. Yeah? So this is basically now has the following problem, that because of the liquid, of course there's movement, but there's also vertical movement. So it's very difficult actually to see the bacteria properly. So 60 times. Yeah. Let's open up the condenser a little bit here. Yeah. So that is basically, I'm gonna turn on DIC now. So maybe we have a slightly better contrast. But basically these are now, so-called rod-shaped, rod-shaped bacteria. They're a little bit longer. Here is the arrow. Yeah, so you see if you look at the individual bacteria over here, they're quite longer, and there are many hundred thousands of bacteria in here right now. And it's okay, but in reality, I don't see them very well. I want to see them much better. Okay, and that's why you want to dry the slide, and why you would like to heat fix it and possibly also stain them, because that is what I'm going to do next. Okay. Yeah. So that is basically simply to show you how it looks like if you put it directly under the microscope. What I'm going to do now, however, is I'm going to show you this whole process of heat fixing. Because what I've done here is I've already prepared, also very difficult for you to see now because of the lack of contrast. I put some bacteria a couple of hours ago on the slide with a little bit of water, and spread it and allowed it to dry up. Okay. Now, the thing is the following. I also would like to stain the bacteria with methylene blue. And there is now the problem is that, it's dry, but the bacteria, they do not stick to the glass yet. I want to bake them to the glass. And I do that by heat fixing the slide. So what I'll be doing is I'll be pulling the slide with the specimen away from the flame. I will be pulling it through the gas flame to heat it up. This will cause the bacteria to form cross links to the slide, and they will stick a little bit like an egg in a frying pan, which sticks. Yeah, and that's important because then I can add the stain without the cells being washed off. Okay, that's why heat fixing is important. So in order to do that, of course, I have to. Now maybe I'm going to do it like this. I do have to. What's the best way of doing this? I have to maybe move, put it like this. I don't know. Yeah, I have to be careful a little bit that I am not able to, yeah, so I won't burn the. Okay, here we go. That's better. So, and so in order to do that, I'm gonna pull the slide. 1, 2. I turn it off, and when I hold it on my hand like this, it should be, yeah, I should be barely able to hold it. It's pretty hot, but it doesn't burn me. Okay, that's then the correct, yeah, amount. So it's quite hot, but not hot enough to actually burn. It does hurt. Yeah. So let's cool it off a little bit. So I'm going to put it on here a little bit to cool it off. And now it's heat fixed. Let's have a look at this now. Okay, and we're going to see that hopefully, yeah, the bacteria are now more easily visible because they're now all in one plane. They're all now spread across on the surface of the glass slide, so they cannot move vertically anymore. And this should make it easier for us to actually see the bacteria. So where is it? The microscope view. So I have to, of course, get everything back into focus again. Okay, here we go. It's still quite dense here. Let's go up with the magnification. No cover glass, by the way. Okay, no cover glass. Yeah, and I think you get the idea that now 60 times. I think you get the idea that, let's open it a little bit, that this is actually much better to see. So yeah, so heat fixed bacteria, they don't move around obviously because there is no liquid there. They're all in one plane, so they're much easier to focus. Yeah, and you're able to see now the E. coli bacteria much better. By the way, I'm not going to use oil immersion right now. Normally what you can do is you can apply now immersion oil. Without a cover glass, you can then use the oil immersion objective. So you're rotating, you're putting the oil directly on the heat fixed bacteria. That is possible. Yeah, I'm not going to do that now. I'm just a little bit lazy. So this is now, yeah, 100 times, but without immersion oil. So it's still pretty good, I would say. Yeah, so I would say it's still pretty good. Yeah, yeah, and basically you see that these are rod-shaped bacteria, so they're a little bit long. Some of them do not all have the same length because what they will do is they will grow in length and then they're going to divide. So, and if you want to see the whole thing in phase contrast, it's also possible, but I do have to switch the filters around a little bit. Yeah. I'm going to, and I have to refocus. Let me see if I'm able to see that. Yeah, it's a little bit too bright. With 40 times magnification is not very high. Yeah, but that would be also in phase contrast. Not so good, especially because I need a slightly higher magnification. Yeah. But this here is with 60 times. So, and what I would say now is the following. Let's do a little bit of staining. How is that? Yeah. So let's do a little bit of staining. And what I'm going to do now is I'm going to take out the slide. And I forgot again some of my tissue paper that I need. And I'm going to now switch back to the desk view, and I'm going to apply a drop of Löffler's solution, which contains methylene blue, directly on the heat-fixed bacteria. Because they're heat-fixed, the stain will not remove the bacteria because the bacteria stick to the glass surface. Okay, so I'm going to simply add a drop here. And I'm going to allow it to stain. Maybe a minute or two. Okay. Takes some time for the stain to be absorbed into the cells. And I need some paper. Oh my gosh. Okay. I need to maybe pull out a little bit of tissue paper from from the trash can. Here we go. Because what I'll be doing now is I'll be I have to now remove the stain again. Now the question is, how am I supposed to do that? I cannot wipe it because then I'm going to probably scratch off the bacteria, right? So I need some way to actually get the stain off without touching the bacteria, and there is an easy way to do that, and that is also the reason why we heat fixed it, because they will not be washed away, and that is simply by holding it like this and using some water and rinsing it off without so you don't want to go with the water directly on the bacteria, but over it, and then you're kind of rinsing away, and you can already see that there where the bacteria were, it's more blue. Let's use a white background. You see, actually it stays blue, because the bacterial cells have been stained. And then what do you do now? You wait again and until it's dry, a couple of minutes. Now, of course, we don't have time to do that because we're also impatient. And this is the reason why you have guessed it, I prepared already a slide, and this one is now dry, so that we don't have to wait. So I'm going to put this slide here away, and we just pretend that this one is the slide that we've made. And I'm going to put now this slide under the microscope. And then, believe it or not, it's pretty much everything I wanted to show you. And then I have a little bit of time to actually go through your questions. So again, as before, that's not the correct filter. It's a little bit too bright. I have to focus on it. You can already see there's something bluish here. Okay, let's go up with the magnification. Let's go up with the magnification. Okay, and 60 times. And you can see that they're all nice and blue, and even 100 times now without oil, without immersion oil. Normally you want to use immersion oil, but you can see that even without immersion oil, it might not be quite as clear and as sharp, but you are able to see that. By the way, this is bright field. I've removed the filter, okay? So I just want to make this clear. So that is basically, these are not stained E. coli bacteria. So see that some of them are still clustered a little bit around. So many bacteria. And over here, as I picked it off from the colony, it did not dissolve all of them yet. So that's why sometimes there are those large clusters. But I think you're able to see quite well that those bacteria have now been stained with methylene blue. So that's basically, pretty much everything that I wanted to show you. I've been doing this now, 48 minutes, 50 minutes. I think that's now a good time also to maybe have a look at some of your questions, because some of you might have been waiting quite patiently already for me to respond. But the reason why I wanted to separate this a little bit is because some people just wanted to watch everything in one session, and then I can go a little bit off topic a little bit with some of the other questions So I'm going to start at the beginning. So I'm going to go, lots of comments again. I'm going to scroll to the top, and if there are any questions that I'm able to answer, then I will do that. So there has been a lot of greetings again around the world. Thank you for that. Okay. I thought E. coli was dangerous. And the answer is it depends. And that's, I think, actually one of the things that I wanted to talk about in any case. So Tini asked right at the beginning of the chat, I thought that E. coli was dangerous. And I say some of them are. It depends very much on whether a bacterium is able to produce certain poisons, toxins or not. And if you've got an E. coli bacterium which is able to produce certain toxins, then they can be dangerous. The ones that we've got over here, that I've got over here, these do not produce those toxins, and that's why they can be eaten. Okay, because so this is yeah. So indeed, sometimes you hear in the media there have been E. coli infections and epidemics and so on. Yeah, sure. And then those strains, specific strains of E. coli, they were able to produce certain poisons that caused people to become ill. Yeah, yeah. Yeah. Oh, yeah. Only certain variants. Okay. Hello from Hungary. Yes. Okay. Yeah, there are greetings at the beginning. Yeah. What is the difference in refractive index? What is the difference in refractive index the same as how you are able to see water in glass, drinking glass? So that's an optical question. When you're, for example, let me put another bacterium over here under the microscope. Okay. Over here, these are non-stained ones, if I'm able to find them. And the question is, why are we able to see them? Why are you able to see those bacteria, the non-stained bacteria? Bacteria are transparent, most of them. Why are you able to see them? And the reason is because of differences in refractive index. And if you've got phase contrast, then you're able to even see them even better. So, so that is the thing. Why bacteria generally are difficult to observe. Because the refractive they're made mostly of water, of course. And if they're in water, then the difference in refractive index is too small, and then you're not able to see them properly. Okay. So yeah, there are again a lot of greetings here. Hello from Canada. The desk looks different today. Yes, I had to set the camera a little bit different because otherwise I could not have showed you the gas burner. Yeah. Yeah, the sound is great. This is essentially from the beginning still. Hello from Netherlands, from Spain, from Switzerland. Sound is good. Hello from Vietnam. I'm probably going to buy Press Science Infinity in a few months. I've seen the Press Microcam Pro 5. Yeah. I cannot say very much about this specific model because there's so many different brands around. Okay. Yeah, cool that you're buying a Science Infinity. I have heard that the model, okay, I cannot say so much about this model because unfortunately I don't know it. Yeah, so I will skip over that. Yeah, 3 centuries of microscopy. I feel we should know more. Yeah, we already do know quite a bit about microscopy, luckily. So the physics of microscopy have now been understood now for quite some time. Yeah. Do you have any recommendations for a microscope ergonomics? Neck hernia, and I'm planning a major, which requires me to use microscopes a lot. Well, I can tell you the following. Microscopes ergonomics means, especially when you're looking through a microscope for a very long time. I mean, some doctors, for example, researchers just spending, 8 hours a day behind the microscope. I mean, that's their job, right? And in this case, ergonomics is really important, especially certain brand microscopes. What they have is you can adjust the height of the eyepieces, for example. Also, for example, very large field of view with a large eye relief. That means where you don't have to go all the way close to the eyepieces. That is also important. The correct height is, of course, very important. Proper chair is important. I would say probably that maybe another option that you might want to consider is to maybe not look through the microscope, but to connect the camera and try to do some microscopy using a monitor, for example. That is also something that some people are using. So there are a variety of things, but I would say that probably an important thing would be is that the microscope has the correct height, and if it is too low, then put something under the microscope to increase the height. Yeah. Okay. Yeah. Hello. I'm new to microbiology. Do you have any books to recommend for beginner? Oh my gosh. There is I don't know if it's for beginner. It's a college level book. It's Brock and Madigan. It's called. It's the authors. But you see, the field of microbiology is so large that it really depends on which sub-branch you're interested in. Yeah? So this is, I cannot recommend one single book really, but there are, you'd have to go specific, more the cell biology you're interested in, or systematics or genetics. There are so many different subfields and it's becoming quite specific very quickly. And that's, I think, also one of the problems here. Yeah. Does the microscope camera quality also depend on the computer quality? Well, the image, no. Let's put it this way. The microscope, the quality of a microscope camera depends on there are a variety of parameters. The resolution, of course, the chip size, the signal to noise ratio. Generally, I would say that these days, modern microscope cameras that you can buy are definitely good enough. But what is important is that the computer, if you want to do some image processing or if you want to do video capture, then of course your computer also has to be fast enough. And in many cases, if you want to do videos, then the USB 2 connection that most computers have these days, or at least most cameras have these days, I would say is a little bit too slow. So you should get USB 3. Yeah? And then you have a somewhat slightly higher speed. So I would say that this is probably one of the bigger bottlenecks these days. In my country, it is very hot because it's summertime. Are there any interesting samples? Yeah, pretty much everywhere where you go, when you have water samples and when there are algae growing, you can try to put that under the microscope. Yeah. Greetings from the Netherlands. I just went to a great exhibition about Anthony van Leeuwenhoek in the city of Leiden. They showed some of the original microscopes. Yeah, that's yeah, in the Netherlands. Also Delft. I think in Delft, he was also living in Delft in the Netherlands. So I think there is also a museum over there. Yeah, yeah. Make a microscopy joke. Unfortunately, I don't know one. I'm thinking about participating in the 300-year celebration. Are you? If yes, I'm curious to see your videos. Yeah, the idea is if you joined in, is anyone should participate. And if you, yeah, if you're able to, I don't know, even know where I wrote it down now, but if you're able to make some videos, here it is, okay? Yeah, and if you put the following hashtag next to it, then this would be a great way to share some of the knowledge, yeah? Do you have findings from the beach? You can collect rocks from streams, only if there is a greenish or brownish biofilm. Those biofilms that you have on some rocks often are cyanobacteria, and they can be indeed quite interesting to observe. Hello from Germany. Yeah, I made my first spore print directly on a slide, and it worked amazingly. I added some 2% potassium, yeah, KOH, calcium hydroxide, and took pics of the spores. There were thousands. Yeah, so basically a spore print works like this, is that you take a mushroom and you put it over a slide, and then you knock on the mushroom, and then the spores will fall down on the slide. Okay, and this way you're able to make a spore print. So I will need to check my fish pond, or maybe I should look at the starch grains. Yeah, there are many things that you can do. Yeah, I have some jars on my desk, but I've not looked at them yet. I've been away for work. So this is also a possibility. I also have got several jars. If you collect some pond water and let it stand, then yeah, often the algae and other microorganisms will start to grow. Yeah. Yeah, so I will jump ahead. What do you think is the scariest looking organism? Well, honestly, if you look at some insects under a stereo microscope, some of them can look quite scary. Yeah, or some yeah, even yeah, some worms and so on can look quite scary, especially if they have certain yeah, mouth parts that look scary and hooks. Yeah, so there. But I would say that these are not microorganisms, but rather small animals that you see differently when looking under a stereo microscope. Okay. So what is your oldest microscope? Not from when you got it, but from when it was made. My oldest microscope is an Olympus CHA microscope. I think it's from the 1980s. And yeah, I got it for free because it didn't work anymore, because I could not turn the focus knobs anymore. The reason I could not turn them anymore is because over the time, the oil in the gears starts to turn hard and solid. And getting it fixed, you have to go give it to a service company who takes it apart, cleans it, re-lubricates everything. And this cost me 200 euros. Simply getting it properly cleaned. Now it works very nicely again. But this microscope, I think, is from the middle 1970s, 1975, 1980s. Yeah, Olympus CHA. Yeah. So did you use dry? I hope you will like. Did you use dry yeast for what? Well, dry yeast is I've also got, okay, but dry yeast is not the same as yeast extract. Yeah, so that's also important to yeah. E. coli at the drugstore, yeah, sure. This one, these E. coli, we got, yeah, okay, yeah, we got it at the drugstore. Yeah, yeah, they help also. E. coli help restore the intestines. What is an intestine? Also, you won't be, you cannot see it now because of the green screen. But yeah, so these are capsules that people sometimes eat in order to restore the digestion again. Yeah. Have you ever gotten hurt in microscopy? Yeah, when I pricked myself to draw a blood sample to put under the microscope. Generally, you got to be careful, especially when you work with microorganisms and so on, that you don't hurt yourself, because this can be a source of infection, of course. But luckily, I never really got hurt. In microscopy, one thing that you should, concerning safety, is important is always make sure that you have proper ventilation. If you use any solvents, for example, you don't want to inhale those. Yeah. Can you safely grow this at home without any danger? Okay. The reason why I've showed you the whole thing with the Petri dishes and so on is because there are so many YouTube videos out there where people actually do stuff that I consider pretty dangerous. Especially, not pretty dangerous, but something I wouldn't do. Look, if you're growing E. coli bacteria, E. coli are known bacteria and they are safe because I know that they're safe. So they're in biohazard category 1. As soon as you start working with unknown bacteria, you already need a permit for the lab, and you're in elevated category number 2. But as soon as you start making fingerprints, or if you start putting stuff on the plates where you don't know what it is, you are already actually in an elevated biohazard category. And you shouldn't do that. The thing is the following. Is it possible to also grow dangerous stuff on these plates? Yes, even especially, for example, if you've got contaminants on it. So when you don't do this properly and you've got a colony growing somewhere, you don't know which bacterium it is, and you have and the number of bacteria, as you can see here on the background here, is astronomically high. So in that sense, it's not even if you are using safe bacteria, if you don't work hygienically, there is of course the possibility that you end up growing bacteria where you don't know what it is. Yeah. So in that sense, I'd be careful even here. So on the other hand, it is of course every time when food spoils, you grow unknown bacteria. But one thing that I would definitely avoid doing is growing bacteria that are able to grow on your body, like your fingerprints and bacteria from your skin or so. I would not do that. Yeah. Because those bacteria have the possibility to grow on your body. Yeah. So yeah. Can I get Petri dishes prepared already? Yeah, Amazon sells them. Yeah. So these are the ones. These are actually ones that were sold for growing fungi. Yeah. And they came in a box, not so cheap. Yeah, but yeah. Well, I tried to grow bacteria on here, and as you can see, it actually worked out. Yeah. I dropped the cover slip on my floor, and it took me over a week to find it. Just use a vacuum cleaner. Use a vacuum cleaner or some broom to cover slips are not so expensive. Yeah, but of course, if they break, then you might get injured. Yeah, there seem to be bacteria everywhere. Yeah, they're pretty much on a lot of places. Yeah, yeah. So as a matter of fact, there are more bacteria growing on our body and in our digestive system than we have body cells. Yeah? So, and I will go down here. Yeah. So this is fascinating. Wow. Which Petri dishes are you using? Plastic, glass? I'm using the plastic ones, disposable plastic Petri dishes. I'm just going to switch over again to, How big are they? I don't know. I don't even have a ruler here. That's basically the standard size. Yeah. Are they 9 centimeters across? Yeah. I would say probably. Yeah. So these are the standard size Petri dishes. Look, I've got also some tiny glass ones here, but I usually use them only for storing water or some other samples, but not for any other things here. Yeah. Yeah. So let me go down again here. That will be a lot of challenge. I should look inside my kitchen to see if there is a Bunsen burner. Yeah, I mean, yeah. I also need some E. coli pills. Don't know if I can buy them or not. You know what you can do is why don't you buy some yogurt bacteria? Because they can be bought over Amazon, and you put them into milk and you can make your own yogurt. And that's definitely safe. And then, by the way, if you take the yogurt and you put it under the microscope, then you can also see. You can even see, some bacteria in chains. Yeah, so it's also quite nicely possible. Yeah. Can anyone tell me what kind of legal consideration should I have if I want to culture bacteria in the USA? I've been in photos where people make them with it too. Yeah, and that's the whole thing. I mean, I've seen on YouTube a lot of people who are actually posting stuff like this, but honestly, you're in biohazard category too. You need a license for that, or your lab needs to be licensed. Yeah. And that's something that's really important. Culturing bacteria. Well, if you buy this here and you make your own yogurt, you're culturing bacteria. If you make sauerkraut at home, you're culturing lactic acid bacteria. So essentially, fermentation processes are part of our everyday life, but it really depends on the microorganisms that you're using here. Yeah? Can you send photos of the parasites, bacteria, fungi? I have great photos. Okay, I think that's meant for someone else. Okay. I thought you said that bacteria were boring, put under the microscope. I think so compared to the others. And also, I don't have an inoculator tube either. Yeah, I personally don't considering the other things that you're all able to see. I personally consider bacteria not so interesting. I mean, look, you're just dots, right? Let's go up a little bit with the magnification. I mean, yeah, that's not so super exciting. Yeah. Yeah, I think, yeah, okay, it's interesting to see them once, but honestly, yeah, that's the reason why I said there are probably more interesting things to look at. Parafilm, yeah, parafilm is where did I put it again? Is the thing that I use to cover, the Petri dishes. It's called parafilm. Here it is. It comes in a big roll, and if you know what, I show you again. I'm just also going to cover this plate again. So you do the following. You cut off a little strip of parafilm. Yeah, and you basically close it off like this. This parafilm is not only good for, you have to stretch it and then it becomes sticky. And yeah, this parafilm is actually also used to cover other things, like, for example, bottles, or I should, look. Why not? Yeah, I've got some some moss here. Yeah, and what you can do is, if you want to, you also take a little bit of parafilm, and look, you can also cover it like this. Okay, and it makes an airtight seal. Yeah, that's basically what it's used for. So it's a general purpose tool, which is kind of fun to use. Yeah, but of course you want to make sure that there is air coming into the moss, going into here, and that's why I'm gonna remove it again. Yeah, it's single use because you cannot use this anymore. Yeah. So the date, of course. Okay. Nice visible colonies. Thank you for the video. Somebody's saying, okay, thank you for the thank you. What is the purpose of using parafilm? I just explained. I work in a microbiology lab, but I've never seen it on a Petri dish. The reason why I use the parafilm on the Petri dish is because, especially if you want to store them longer, it prevents evaporation of the liquid. Yeah. Or what you can do is you simply stack them together and you put them into a plastic bag and then you put it into the refrigerator. That's also possible. But also for transporting, for example, the Petri dishes, it might be useful. And I also use it for safety. So essentially, when it falls down, that the lid cannot go off, and contaminants are prevented from actually going in. That's it. I have not used, while I was at university, we did not use parafilm on the Petri dishes generally, only occasionally when it was necessary. Yeah. So because it does take time to wrap everything. Yeah. So let's move on here. Yeah. So how do you safely clean the object glass afterwards? Generally, what do you do with those Petri dishes once they're not used anymore? They go into an autoclave. This basically means into a pressure cooker. And they're being autoclaved for about a half an hour at 110 degrees or 120 degrees centigrade, and this kills off everything. Of course, the agar medium is going to melt as well. And if you don't have an autoclave, you can put hydrogen peroxide over it. That's probably a little bit risky if you're not careful. Hydrogen peroxide is very effective in killing microorganisms, but some microorganisms form an enzyme called catalase, and then they will convert the hydrogen peroxide into oxygen, and there's a lot of foaming going on. So you have to be a little bit aware of that as well. Otherwise, it might start to foam quite a bit. Yeah. So staining bacteria. Just a second. Oh, I kind of lost where I am. Where can I find E. coli? Naturally, you can find it in the intestine, but in this case, I got them over those capsules that I bought. Or you can order them, or try to order them from a cell culture organization, but they might not send it to you because you're not a laboratory. Okay. I majored in biology way back in the 70s. Your videos bring back memories. Yes. Yeah. Doesn't methylene blue kill the bacteria? Yes, it does, but they're actually already dead because they're heat fixed. Yeah. Is it not used for throat or infections? Yes, methylene blue can also be used to treat certain infections, and also methylene blue is also used in some aquariums to treat fungal infections of fish. So if the fish become ill, then you can add some methylene blue into the fish tank, and then it also becomes blue, but this might actually help the fish. I've seen a video with 100 times phase contrast with and without oil, and I've seen that no oil drastically affects phase contrast. Yes, you need oil for resolution. I have not used oil now because I didn't want the mess, okay? But it still worked. Yeah. Could you show the freeze-dried E. coli powder under the microscope? Okay, why not? So the question was, can I not put this one directly under the microscope as well? Yes, but I'm not quite sure whether we're able to see quite a lot. I've not tried it, but that's actually one of the things. Why not? Okay, so why not try it? So I'm going to take again a little bit of water. Put some water here, and I'm going to do it now a very cheap way. I'm going to dip it directly in here because I'm going to dispose of it anyway. I'm going to transfer some of the bacteria here now into the water. This was a very unprofessional way of doing that. Okay. I'm going to put a cover glass on top, and let's have a look directly. I assume that there are going to be a lot of clusters. So I don't think that it will look quite as nicely as the other one, but who knows? I always have to check. There were two cover glasses. So let's have a look. Okay. I might remove a little bit the excess water from the side here to make sure it's really flat, and so that the bacteria are not able to move too vertically. And let's have a look. Okay. Thank you for the recommendation. I don't know how it will look like. Let's remove the old slide. 2 old slides here. Up, I dropped it. Doesn't matter. So let's put in the slide directly with the E. coli freeze-dried powder. And let's focus again. It's a little too... I would assume that maybe there are lots of clusters present. But maybe. Let's go up again. So let's ignore the clusters, but let's try to put DIC in. Maybe the DIC makes it a little bit better to see. I need a little more light. Yeah. Yeah, so that's basically the powder directly, and you might be able to see those dots in the background. Yeah, these must be, yeah, these are the E. coli cells. And they're kind of wiggling around a little bit. I think this is due to Brownian motion. Okay. So just because tiny dots move around does not mean that they move on their own. Yeah, I think this is due to Brownian motion. And because, yeah, they look actually much smaller than the others. And I think maybe because they shrunk together because they were freeze-dried. They're freeze-dried. Maybe they will, as they absorb water, maybe they will become a little bit larger. And it does take them some time actually to start dividing. And then maybe they're going to take on their original size again. I don't even know if you're able to see it on YouTube in a live stream very well, if the image quality is good enough. Yeah? I mean, I could try now phase contrast. Let me try phase contrast 40 times, but it's still a little bit small, I would say. Yeah. Yep. The liquid is spreading now. Yeah. So all of those tiny dots in the background, these are the bacteria. So again, this is directly the powder, and yeah, not very impressive. Yeah. What is refractive index? I do not know how microscopes work. Oh, that's a long topic. Yeah, basically, refractive index is how certain transparent objects like glass and water, how they bend the light. And the more they're able to bend the light, the higher the refractive index. Okay, so this is a little bit of a, yeah. Which other stains could you have used? Yeah, what you do is traditionally in microbiology, what you can also use so-called gram staining. And this is a little bit more complex. You can use iodine for staining, but I would say that methylene blue is a very common stain because it's a very general purpose stain. Okay, but I think I'm just going to put again the other one in here. We have something to look at. So there are a variety of different stains. I've used, I don't know how many of them already. You can stain for spores. You can stain for example, for other inclusion bodies and storage substances. Iodine works as well. So it depends really what you want to make visible. Yeah? Staining biological matter is not usually done by a standard nail. It's to do with the environment and invasive species, tissues and concentration. Send a sample is not a good idea. So that's basically the following. Is what happens if you want to send a sample from one country to another country. Sending it is easy. You just put it into the mail and send it off. But there's a problem. Even if you're able to receive a sample, if you want to work with a sample and if you want to publish your results, then basically you have to be able to document how did you get the sample. And if you are kind of doing this in an unauthorized way, then you cannot publish your research results. Just sending it alone is, of course, the easiest thing. Yeah. But if you wanted to research with it, then the whole thing has to be approved, right? And for this reason, if you want to send biological matter, there's a whole list of criteria that you have to meet, of course, yeah. Here are basically a whole bunch of stains, yeah, mentioned, yeah. The cat and the daughter have blackish worms here like that will not die. I can send photos. Well, I would say if you have problems, it's generally an issue with parasites in your house, pets, and so on. I think it's important to get it sorted out, what it is, and then you want to talk to a veterinary doctor. Self-diagnosis is never a good idea, not on yourself and also not on your pets. And I would say if there are any certain concerns, then I would definitely talk to specialists. Have you ever done fluorescence microscopy? Yes, at university. And a little bit also at home, experimentally, where I tried to make things glow by shining a UV flashlight on the sample. Yeah. I have a dermatologist friend and I was gonna try and get a Botox sample, but I don't think that's worth the risk. Botox is a very strong poison. Also, I think not very stable, so you have to make sure that it's very stable, and I wouldn't mess around with things like this. Yeah. The highest level of biohazard is category 4, to my knowledge. Biohazard level 1 is safe. You basically are working with known organisms and they do not cause any problems. Biohazard level 2 includes unknown organisms. Yeah. Biohazard level 3, for example, AIDS, HIV. The HIV virus is part of biohazard level 3. Basically means that there is no cure for the disease, but it's not very infective. So you don't get it so easily. Yeah. But once you get it, there is no cure. And biohazard level 4, Ebola, for example, Lassa virus. Those, these viruses are very infectious. You can get them easily, and the mortality is also very high. So these are basically, if you want to know how biohazard level 4 looks like, then the people are working in spacesuits. So there are not every country has a biohazard level 4 lab. And biohazard level 3, I've once visited a biohazard level 3 lab. There are several security doors, and the lab is under pressure, so that if there is a leakage of air, lower pressure. So if there's a leakage, then air is going to stream into the lab and not out. Yeah. So there's even a air pressure difference. Yeah. What about mouse bacteria? Oh, there are plenty of them. All you do is you take a saliva sample and you put it under the microscope. However, you're able to see them, right? However, sometimes there's so much other stuff there as well that it might be a little bit difficult to actually distinguish what are now bacteria and what are your own cells. Yeah. Can you share any information about the Shiga from Shigella toxins and how to deal with them? I don't know much about that. I would say, call this massive diarrhea. I don't know much about the biochemistry, but the idea here is just like with other toxins that bacteria which are able to produce this toxin are causing the problem. So it is not the bacterium as such always, but sometimes also the toxin or the substance that they actually produce. Yeah. So do I need a license to make compost then? That's a good one, right? When you are making compost, you are essentially growing an incredible amount of unknown bacteria. When you have some food spoiling in your refrigerator, you're growing an incredible amount of unknown bacteria, and it's all legal, right? So that's the thing. But if you are a laboratory and if you want to grow bacteria in a petri dish and so on, then it's a different issue. Yeah. And do you make fungus smears in the same way as bacterial smears? I've not grown some. Well, let's put it this way. I think that would be possible. I guess, especially if you are spreading the spores. However, fungi have the tendency to spread very quickly. The mycelia, the hyphae, they radiate very quickly. So if you've got a fungal contaminant over here, it's quite easily possible that within a short time it's able to spread over the whole plate. Yeah. That's a little bit the problem that I've seen with some certain fungi. Yeah? But I think it can be a little bit more difficult to kind of spread the fungi because of the hyphae, and then it kind of spreads very quickly. Yeah? Are you using LB broth? No, this is not LB broth. I think this is I think it is the medium according to Kimmel. Kimmel, I think, if I remember correctly. Yeah, I was not able to find LB broth ready-made, unfortunately. Yeah. Okay. So I'm going through the comments very quickly. As a side question, do you know if makeup kills Demodex mites? I don't know about that. Seriously, I don't know about that. That's an experiment that you might want to try. You can isolate the Demodex mite and add a little bit of makeup. Yeah. Have you ever bought a cheap microscope that way but it's better than expected, or an expensive microscope that was way worse than you expected? That's an interesting one. Let's put it this way. You do get a little bit what you pay for, but I would say that from the optical quality, the microscopes were not so different, because sometimes the optics were fine, but usually the quality difference came usually from upgradability, or it came from ergonomics, as we already talked about, or from modularity and so on. So I would say that essentially, or maintainability, essentially, it depends also quite a bit of what you really mean with better or worse, because different microscopists have different expectations really to the microscope, and it is not always the image quality that counts. I once, many years ago, bought a stereo microscope, and I was rather disappointed. Was very nice quality when I looked through it, but I was kind of very disappointed with the pictures that it produced when I connected the camera on top, because some of the optics did not work very well with the camera optics of the adapter. Yeah, so sometimes it's also a combination of different factors that determines the final quality. Yeah. How are the Dilution's Earth slides? Yeah, they are still in my cupboard drying. And yeah, they are essentially, they look pretty much like on the first day, I would say. Yeah? Is it true that two microscopes can have the same optics, but one can be more expensive because of more features? Of course. Yeah, of course. As a matter of fact, this is definitely the case. Often microscopes are manufactured by the same company and rebranded, and sometimes you even use the same parts. And so sometimes there are not even that many different optics, and but they're kind of reused on different microscopes, and they basically, the microscopes differ, for example, in the features that they have. Usually even the features are not so much different from each other, because the standard microscope has all of the same features, a condenser and a coarse and fine focus, mechanical stage, and so on. But I would say that some microscopes you can connect, let's say, 5 objectives instead of 4, right? If you want to consider this as an additional feature. Back on topic, once you have the cultures completed, how do you dispose of them? Well, I have to basically autoclave them, so that means I'll put them into a pressure cooker for half an hour, and then I can dispose of them in the regular waste, because everything's killed off. So I think I'm pretty much through with all of the questions, and I've been out live online for 1.5 hours now, almost again. If you joined in a little bit late, I just wanted to give you a short summary of what I've done, I've basically been growing. I've been growing bacteria from a freeze-dried culture. Okay, and basically I showed you how to prepare and how to streak them on a plate. And then I put also some of those bacteria under the microscope. I showed you how to make a so-called heat fixed slide. You can see it again here a little bit, by suspending some of the bacteria in a drop of water, allowing it to dry, and then heating it up over a gas flame. And then I was also staining it with methylene blue, and that's basically when we were able to see the different bacteria. Okay. So the next, just some kind of remaining questions. Are you going to do one tomorrow too? I would say, probably not this time. I know that I've been thinking about doing a shorter live stream on a Saturday, and a shorter one, also the same one on Sunday. But I think, I'm running out of, otherwise I'm starting to run out of plates and of material as well. But what I will be doing is if you have any recommendations concerning any topics that you're interested in, please inform me about that. And there's a few remaining questions. Isn't it enough to use 75% alcohol to disinfect? Better than nothing, but generally this is not what's done standard-wise, because don't forget one thing. We have billions and billions of bacteria here, and even if we start to kill off 99.999% of the bacteria, we still have many bacteria left. So So what we need is we need a very effective method that kills pretty much all of them, and simply going with 70% alcohol might not be enough. Yeah. Okay. Yeah, thank you so much for all the information. Thank you so much for, okay, there are lots of thank yous here right now. Okay. What's the largest objective you've ever seen? The largest microscope objective, I mean, they're pretty much the size, maybe. Yeah. Sometimes it's not only the size, sometimes the diameter is quite large. Yeah, that's, yeah, I would say that the size also does not always tell something about the image quality. Yeah. So what I'm gonna do is the following. Because I'm also starting to become a little bit tired right now. I'm going to leave it at that. I hope it was interesting, and I hope that you got a little short insight into some bacteria and basic microbiology today. Yeah, in summary, not necessary if you're interested in microscopy to do these things, but I just thought that it might be interesting in any case. Yeah. So I would like, yeah, it's time to tidy up now, all of the mess that I've got here. And I want to wish you all the best. Happy microbe hunting as always. Do stay tuned to the channel, subscribe to it if you like these videos. I'll be posting, of course, I'll continue to post videos, not only do live streams, but I'll also post videos. And I wish you all the best, okay? See you around next week. Bye-bye. Bye-bye.